Hi everyone. Gene cloning refers to introducing a desirable DNA fragment into a living host cell where it stabilizes, expresses, and multiplies as the cell divides. Cloning vectors may be either plasmid based, virus based, or artificial chromosomes. The prerequisites of a good cloning vectors include: they should be small in size so that it's easy to handle them, plus they are able to enter the host cells without any difficulty. They should possess an origin of replication so that they don't need to integrate into the main genome, thereby not disturbing the housekeeping functions and never causing fatality to the host cell. Additionally, they should be easily able to get transferred from cell to cell. It should be easy to isolate them from the host organism whenever required. They should contain at least one, and desirably as many as possible, unique restriction endonuclease sites for cloning of foreign DNA. It's even better if they possess a suitable MCS or a multiple cloning site, which is a synthetically constructed bunch of many unique RE sites. Then, good cloning vectors contain at least one, or if possible, multiple selectable markers. For example, AMP or ampicillin resistance gene for prokaryotes is a selectable marker. A powerful feature that a cloning vector could have is to possess an RE site within one of its selectable markers so that the possibility of insertional inactivation is there. It means that if cloning is done using the site, the marker should get shattered or inactivated, though it was intact and functional prior to the cloning event. This strongly helps in identifying the proper clone, that is, the screening and selection of the recombinant colonies of transformed cells. In this video, we are discussing the simplest plasmid-based cloning vectors for E. coli. pBR 322 of the pBR series, named after its makers, Bolivar and Rodriguez, has been the hot favorite of researchers through and through. It is an artificial or man-made plasmid vector with a size of 4363 base pairs weighing 2.9 megadaltons. It possesses about 21 unique RE sites as well as genes of ampicillin resistance and tetracycline resistance as selectable markers. PST1, PVU1 and SCAV1 sites lie within the AMP resistance gene, while BAMH1, SAL1, EXMA3, EcoR5 and some more lie within the TET resistance gene, giving rise to a possibility of insertional inactivation, meaning that if the foreign DNA is inserted using any of these sites, that particular marker or gene will get shattered. This would manifest in loss of resistance to the antibiotic it encodes. Besides these, other unique RE sites positioned outside these markers are EcoR1, Henry3, BAL1, NDE1 and many more. pBR 322, being of around 4.5 kb size, can accommodate an insert of up to 5.5 kb as the maximum size considered suitable for entering and transforming a host cell is considered to be 10 kb. Here, small p stands for the fact that it's a plasmid, B and R stand for its creators, Bolivar and Rodriguez, and 322 is the derivative number of the series. Although it was designed for use in everyone's favorite prokaryotic host E. coli, but it was seen to be compatible for use in some related species such as Escherichia marcescens too. pBR series was constructed from three natural plasmids of E. coli, namely PMB1, PSC101 and RSF2124. Another famous member of the pBR series is pBR 325, whose size is bigger, it's around 6 kbp, and besides AMP and TET resistance genes, it also has chloramphenicol resistance gene, also offering the possibility of insertional inactivation of the third gene chloramphenicol resistance for the purpose of robust screening and selection purpose. However, the size of the insert it can accommodate is only 4 kbp. Based on this pBR series, scientists of University of California came up with the pUC series, or popularly known as pUC series, where UC stands for University of California. This was done with an intent of providing a great alternative to the pBR series. This vector is much smaller, that is 2686 base pairs in size, and is much more relaxed by nature, that is exists in much higher copy numbers per host cell. Also, it dishes out a more reliable and interesting blue-white screening and selection system. Similar to pBR series, pUC vectors also contain the AMP resistance gene. In addition, they possess a LAC-Z- gene also, which is carefully designed to get insertionally inactivated by virtue of cloning within this gene. This results in the formation of white colonies on a culture medium containing gratuitous inducer of the lac operon IPTG and chromogenic substrate XGAL. In contrast, if this LAC-Z- gene is intact, it produces blue colonies. This may be attributed to the successful expression of this gene, producing a functional beta-galactosidase enzyme induced by the IPTG provided, that in turn convert the chromogenic substrate XGAL or 5-bromo-4-chloro-3-indolyl galactoside into a blue color product. This kind of differential screening and selection of pUC vectors is much better and highly reliable than that of pBR series. However, a strict prerequisite while using this cloning vector is that a special E. coli host cell is required that contains the usual lac operon, but its LAC-Z portion is deliberately made non-functional. So, if the unshattered LAC-Z from pUC is provided or available, the alpha-complementation of the protein products takes place and blue colonies of E. coli appear on the selection medium, else they are white. Researchers screening for the recombinant colonies containing the target DNA look out for the white colonies, since the blue ones would contain just the native vector rather than the recombinant vector. We hope, guys, that you could comprehend our simplified description of some common plasmid cloning vectors of E. coli. Please feel free to share with us your queries, if any. We thank you for your encouraging remarks and appreciation of our work. See you very soon with bacteriophage-based prokaryotic cloning vectors in our upcoming video. Till then, keep learning. It's bye for now.